to us about the role of different types of interneurons in controlling the function of microcircuits or circuits in the cortex.
So today I would like to talk about anyway, we are very interested in the microcircuit of the cortex. And this cartoon just showing the very brief structure of the cortical microcircuit. And we know that the main projection neuron in the cortical cell showing the gray, and also different colors. So I showed that every different kind of interneurons. And then today's my main topic is today's title is Functional Effects of Distinct Innervation Styles of pyramidal cells by Fast Spiking Cortical Interneurons. And then especially I'd like to talk about this connection. Fast spiking basket cell inhibit the layer 5 pyramidal cell. Actually, I published that paper very recently in the eLife. So if you are interested in, please read through that paper. This is just a laser confocal microscope image with the programming in the reaction with Taiwan mouse sections. So the green is the Taiwan positive YMP positive Taiwan cells, and then red is Taiwan positive interneurons. So this part is enlarged here, and you can see Taiwan positive layer 5 cell is surrounded by many Taiwan positive terminals. So I'm going to introduce the functional significance of these connections today. So inhibitory circuits regulate cortical activity and development and plasticity. And a part of inhibitory function is associated with pathologies including epilepsy and autism and schizophrenia. So inhibitory circuit is very important for our higher brain function. However, mechanisms controlling inhibitory static actions are incompletely understood. So I would like to investigate that, and then to do so, I found, first of all, I published this result in this paper, and the inhibitory substance innervate different parts of the target neurons. We inject the myocytin using the wholesale neurophysiological slice recording conditions, and we record these 9 different interneurons of the cortex. This is late spiking neuroglia form cell, and 2 fast spiking basket cell, and then 5 descending axon cells. Some of them we call bldg cell, and one is like a marginal cell. It sends axons to the layer 1. Presumably they innervate the tuft dendrite of the pyramidal cell. And what we found was the cortical inhibitory interneurons, they innervate 3 different structures. One is soma, and then second is dendrite, and also third one is spines. So this red one is a presynaptic terminal of these interneurons. And then spine head, you can see this guy innervate spine head, and then most of the case, this spine head also receive excitatory shown in blue. And then maybe this is just somehow just common sense. The inhibitory non-pyramidal cell tend to innervate pyramidal cell soma, but not distal one. Maybe this is like an older idea, but that is actually not true. This shows you the proportion of these interneurons target structures, shown in soma in black, and dendritic shaft is gray, and spine head is white. So you can see somatic innervation is done by late spiking neuroglial cell and fast spiking cells, and also some of the double bouquet cells. But these guys, they don't have any, especially the marginal cell, they don't innervate soma at all. But their main target is like dendritic shaft. About half to 1/3 of the target is dendritic shaft. And surprisingly, about half to 1/3 is spine head. So in terms of the number of the target structures, the soma is not the major target structures, but dendrite and spine is target structures. So we would like to know the different function of those different innervations. And then also, very interestingly, the Massimo Scangiani published a Nature paper that is equalizing excitation and inhibition ratio across the pyramidal cell. He used a slice preparation, and then he manipulated layer 4 cell expressed channel rhodopsins. So when he just irradiate the laser into this slice, and then he touched the 4 different pyramidal cells. And then he found the ratio of the iPSC and the EPSC is always the same, no matter what the size. So this is their finding. That means like some of the pyramidal cells receive the larger inhibition, but some of the other pyramidal cells receive only small inhibition, some of them. And then Isola Zortes also published a neuron paper, and then this is hippocampus, and then there's like a superficial layer and deep layer of the pyramidal cell layers. And then he found always the superficial layer received, he actually make one spike of the pyramidal basket cell, and then he tried to find the iPSC of the pyramidal cell. And then he found superficial layer pyramidal cell, they receive smaller inhibition compared to the deep part. And this is true of the septal CA1 or temporal CA1. So the inhibition is quite different among the different pyramidal cells. That is already known. So we wonder what's the mechanism. Then we did this experiment. We use a pair recording of CCS pyramidal cell and also fast spiking basket cell in layer 5. And then we try to get one subtype of layer 5 pyramidal cell. Basically, we know that there are 2 major layer 5 pyramidal cells. One is we call CPN cell, and the other one we call CCS cells. And the CPN cell corresponds with a rich tufted layer 5 pyramidal cell, and the CCS pyramidal cell corresponds with slender tufted layer 5 pyramidal cell, which already proposed by the, I believe, Bart Zuckman many years ago. But we would like to just pick up only one type, that's CCS cell. How we find the CCS cell, that is, we inject the dye into the contralateral striatum. Because we know that this CCS cell projects the contralateral striatum as well as the other like ipsilateral striatum. But the CPN cell, they only project the pons and the ipsilateral striatum, but not the contralateral striatum. So we distinguish. This is the method to distinguish CCS cell with tracer injections. And then after the different tracer into the pons and also contralateral striatum, we found this kind of like retrograde model layer 5 pyramidal cell. The red is, we believe this is CCS cell, and then blue one, we use blue tracers, and then these are the CPN cells. They are completely separate proportion. And then I would like to just introduce our method of the slice preparation, slice recording. And a single spike was induced in the presynaptic fast spiking vascular cell. And then iPSC, the response of the postsynaptic CCS pyramidal cell was recorded at minus 65 millivolt holding potential. And then electrode solution, this is a little bit sort of our tips. Electrode solution contained high chloride, 90 millimolar, and then the iPSC is inward current measured. And then usually the internal solution of the electrodes contain 4 millimolar chloride or maybe 10 millimolar chloride. Not so large, but we use this high millimolar chloride because we can get larger iPSC. And so that's the advantage, we thought. And then we did equilibrium potential recording, so we shifted the member potential and how the equilibrium potential of the gamma looks like. And then we found minus 2.5 millivolt is the equilibrium potential of this configuration. So we hold the member potential minus 65 millivolt, and the equilibrium potential of the gamma is minus 52.5 millivolt. So IPSP driving force was 12.5 millivolt. So this is another important data for the data on we do the simulation analysis. And then after the recording, because we have bioscience in the electrode solution, so we did histology to do the to stain the pre and post-synaptic neurons. And then we did DNA reaction, and we showed very nice histology structures of the dendrite and the axons. And then we traced all of those dendrite and axons using the neurolucida to find out the synaptic contact portion of the fast spiking basket cell terminal to pyramidal cell dendrite. So then I would like to just, after the 3D reconstruction, we did EM study to find out the synaptic connections. I would like to show you examples of the pair recording. This one is, I believe, CS28. The number of the experiment is CS28. So the first time basket is a spike, and then the post-synaptic pyramidal cell get iPSC like this, and we know that this is really fast spiking, and this would be typical regular spiking pattern of the pyramidal cell. And this is the reconstruction image of the pre and post-synaptic cells. The fast spiking cell is shown in blue. And then these are the red shows the contact of the fast spiking basket cell terminal to the pyramidal cell. And then this pair only shows the somatic innervation and also proximal part of the dendrite innervations. And it's shown in the dendrogram here. We just count this cell as 4 somatic input and also another 5 dendritic input. But also we found some other pair recording the different morphological feature. That is this pair, CS45, they have only dendritic innervation but not soma innervations. And then if you compare the size of the iPSC, then it's much, much smaller. And then this is actually a summary of the pair recording. This is, we did 10 pair recording. Actually, we did a whole bunch of different pair recording, but we successfully collect the data of the neurophysiology / recording, and also histology, and also neuroregister reconstructions. And then we succeededly get the data from these 10 pairs. And then in first 3 pairs from the top, we found somatic innervations, shown here. Maybe this doesn't work, but it's okay. We found CS55, 3 contact site, and CS28, 4 contact site, and CS56, only 1 contact site. And but the other pairs, we don't see any somatic contact, but only dendritic contact. Good. And also we plotted this bar, it's the actual contact site, and we just plotted from the distance from soma. So, and then this is IPSCT. These 3 pairs showing the somatic contacts, they have a larger IPSC. But the dendritic synaptic contacts, they are not large, they are small. And then this asterisk shows the contact site which is closest to the soma. And then you can see this, and this is really like nicely inversely correlated. Like distance from soma is nicely inversely correlated with the IPSC size. And then we found anyway, we found that distance from the soma, from 33 micrometer from the soma, we can get line here. And then, although these 2 pairs, we see the morphological contact, but we don't see any IPSC. And then we can just make a line here. And then wherever we found the contact site close to the 33 micrometer from soma, we can see IPSC, but these are not, we don't find it. But this tells us the IPSC doesn't contact a lot long distance, only 33 micrometer or so. Oops, sorry. And then we actually did EM study of these CS55 and CS56. If those like contact site confirmed by the light microscope is really showing the synaptic contact or not using the EM reconstruction. And then this is actually the light microscope, light micrograph of the CS56 pair and CS55 pairs. And then this is the reconstruction using the neuroregister. And the blue is the pyramidal cell, and red is fast-spike basket cell. And the pink shows the axon terminal fast-spike basket cell. And unfortunately, because of this slice using the 300 micrometer thickness, the apical dendrite of CS56 and CS55 is we missed because of the the slice preparation. And then actually we realized that this CS56 surrounded by fast-spike basket cell terminal, but a comparison to the CS55, it's a sort of small number of the axon terminal of fast-spike basket cell surrounded. This is much many fibers surrounding the pyramidal cell, post-synaptic pyramidal cell soma. And we did short analysis, and then this is really true. So, and then it's turned out the nice correlation of the size of the IPSC. It is small, but this is large, very large. And then we assume that maybe number of the somatic synaptic contact is a big issue for this IPSC size. And actually, this is the max IPSC because this is average. Of course, like IPSC, the structure itself. And then I would like to show you how we did the EM study. So this is the post-static pyramidal cell of the CS56 pairs. And then this is the dendrogram of the basal dendron. Somehow we didn't see any septic contact onto the apical dendron, but only basal dendron. And then we found one presumable contact site on the soma, and then 1, 2, 3, 4, 5, 6 presumable contact site of the dendron. And then we really want to know if they are really showing the septic contact. And then for instance, this is C1, named C1 segment is shown like here, and then here, and we capture the image like micrograph like this, and then we did this part, and then EM3 serial cells, serial sections, and then so they are the serial sections, and then showing really this part. So with this serial electron micrograph, we did 3D reconstructions, and then the image is like this, and then it's quite same, right? And then we really pick up the right dendritic position for the 3D reconstruction. And actually, this part is C1, and then we found that there is no septic contact by this first particle steps now to dendritic of the post-septic pyramidal cell. But this guy, they got septic contact to the other property. And then I will show you I found the steps, that's it. In CS56, we know that we counted this axon goes probably contact to the soma here, but we count this with one contact site. But the axon actually really goes like this, and then we really don't know what's going on here. And then we did reconstruction of the electron microscope, and then we found 3 steps as really contact onto the soma like this. This is 3D reconstruction image, and then you can see this is quite same image, and then we turn it to the 90 degree, and then we found these 3 steps contact really successfully connected. And then these are the septic junction sites. And also this is another case. This is actually, we didn't count any under the light microscopic observations. But because it's really complicated, we don't know what's going on here. But it's turned out, I can see the substrate here, and also substrate here. It makes a dendritic steps here, and also spine steps here. And then this structure is really combined of the presynaptic terminal and postsynaptic spine head. And also, this is the CS55 data. This is CS55 postsynaptic pyramidal cell soma, and you can see the pointed out by the number, the arrow. This is we found the septic contact by the EM study. So actually, we really don't know it is really contact site or not, just by the light microscope observation, but rather it's just a crossing position, only crossing position. But we have to investigate using the electron microscope to find out if they are really septic contact. And then this is another case because, for instance, this axon goes through like this, but we don't know if this is really connected or not, and we really don't know what's going on here. Because light microscope, this densely stained soma is just really a sort of shadow, and then we really cannot guess what's going on here. I mean, if they have a septic contact or not, and then also this area something going on, but we really don't know. So we anyway found 1, 2, 3 contact sites. When we took a look at the EM 3D reconstruction, we actually did 3D reconstructed soma entire structures like this. This guy actually cut into the half because we do when we do the histology, we resection the slice section into the 50 micrometer thickness resections. And then somehow this guy cut into the two different sections. So I have to do the reconstruction separately. But anyway, we did. And then finally we found these unknown part. We found some synaptic contact. And then we found 13 somatic synaptic contact. So this is a summary of synaptic contact site. CS56, we found 4 somatic contact and also another 4 dendritic synaptic contact. And then CS55, we found 13 somatic contact and also another 9, I believe 9 dendritic and spine synaptic contact. And then because we did 3D reconstruction of the synaptic structures, so I can measure the synaptic junction area quite easily. So we measured, and then we found these are somatic synaptic junction, and then they are very large areas. Like this one, 0.35, and these are the 0.2, around 0.2. But in comparison to the soma synaptic junctions, the dendritic synaptic junction, the spine synaptic junction is so small. They are like 0.1 or 0.5, that range. And it is also same. We found in the CS55 pairs. The somatic synapse is really large, but the dendritic spine is a range of like 0.05 square micrometer or 0.1 square micrometers. So this tells you like a somatic inhibition is powerful than the dendritic inhibition, and that makes me think about maybe the conductance of the synapse is different. And I would like to show the summary of the synaptic contact site in this one, in this graph. So I would like to just mention about this one because of this asterisk is showing a very nice inverse correlation between the site of the contact site and the IPSCPT, but I really don't like this one. So we also investigate this CS54 for the EM reconstruction studies. And also, we believe we must investigate these pairs under the EM because we must know if these contact sites like microscopically confirmed, if they really show the synaptic contact. And then we did totally these 4 pair neurons using the EM, and then we found this. So we found like 13 synaptic contact and the other dendrites and spines innervation of the CS55, CS54, 4 somatic contact and 4 dendrite contact. And then also we confirmed this CS23, at least these 5 are really synaptic contacts. So they are physically make a synaptic contact, but they don't, the IPSC doesn't conduct to the soma. Now I know the mechanism, but this is still another investigation, and I cannot show the data. But also, we found this guy is not really make a synaptic contact. So this doesn't count on this. So like this. And now I can show the reverse relation of this morphology and also IPSC size. Now, I would like to just make an estimation of the conductance of each synapse. Total just nicely showed his paper in Neuron in 1997, the post-synaptic GABA receptor alpha-1 subunit is very nicely correlated with the size of the synaptic area size, like this, using the colloidal gold post-emitting linear reactions. And then very recently, that is 2012, also Zori just introduced us the Nature Neuroscience papers, calcium response of excitatory structure of CSV pyramidal cell. And then so she just stimulate different spines, and then sometimes like calcium response of each spine get activated, but other time doesn't. And then also the first of also I forget the name of her, but she just also showed the number of docked vesicles using the EM structures, and she found this number of the docked vesicles is very nicely linearly correlated with also again area of the synaptic junctions. And also the calcium channel is very important to get this calcium response. And also again, she used the SDS structure method using the CAD 2.1 calcium channel immunochemistry. She found lots of calcium channels. And then also this number of the gold is nicely correlated with the area of the synaptic junction. And also, synaptic junction is very nicely linearly correlated with total calcium response. And also, this is release ratio of the these steps is very linearly correlated with the junction area size. So that tells us the number of the post-synaptic receptors correlate linearly with active zone size. And then the release probability sites are correlate linearly with active zone size. And then we can assume that the synaptic current probably correlate with synaptic active zone size. So the measurement of the synaptic junction is very important to find out the power of the each synapse, IMBB. And then we compared some synaptic junction area and maximum IPSC amplitude. We did like this. Actually, this theoretically, we got this IPSC sort of fracture. And this is the area IPSC of CF56, and this is max IPSC. So we measure the electric charge of this part of the max IPSC, which is I think this one, 325.1 pentacoulomb. And then CS56 is 1,000 pentacoulomb. And then because we know this CS56 has 4 somatic inputs. And then we don't have to think about the other dendritic synaptic input because they don't conduct their synaptic current to the zone. And then we calculate unit electric charge per square micrometers, and that is max IPSC electric charge divided by total somatic synaptic junction area, and we call this unit IPSC electric charge. And then this is the result. CS56 case is a unit IPSC electron, unit IPSC IPSC is 343.3 pentacoulomb per square micrometers. And the CS55, although the size is different, but it comes to a very similar value. So I believe our method, our concept is not, it's true. So this parameter was very similar for the two connections, suggesting that the synaptic current is well correlated with synaptic junction areas. And then we use these unit IPSC to calculate each synapse junction. Anyway, IPS electric charge of each synapse was calculated with the junction area multiplied with the unit electric charge, because we know that junction element. And then the result is this: the electric charge. For instance, biggest somatic contact of CSPC6 is 120 pentacoulomb. And then the spine snaps is only like 20 or 15 pentacoulomb. They are pretty small. And then because of we have models, authentic model cells of CSPC6 post-synaptic grounded cell, and then we know that this electric charge, and then also we know the trace average shape of the IPSC at the zone. So we simply estimate the IPSC conductance of each synapse of the CS56. And then the conductance we estimated is the biggest somatic synapse is S1 is 0.71 nanosiemens, but the small dendrite is like 0.2, and then other spines like 0.1. So the conductance must be very big difference among the different synaptic structures. And then we did simulation study using that result. Now somatic synapse junction is really large, so should have large conductance. Then EPSP should be effectively blocked. So this is our hypothesis. And the dendritic and spine synapse is small, then small conductance, and then EPSP nearby can be blocked. Only nearby can be blocked. So this is our hypothesis for the simulation analysis. So this is how we made a simulation. That is, we know the dendrite structure using the neuroluscular tracing, and then we know we observe every different segment of this grounded cell for the EM reconstruction study. So we know the area of the dendrites, and then I'm not going to tell you the detail about the principle of how the dendrite, the size, dendrite dimension is determined, but I published one paper which really precisely tells the principle of how dendrite size determines using with the non-pyramidal cells in the scientific report paper in the 2011, so please refer that paper. But we found it's exactly the same principle of the dendritic dimensions. So all of those concepts we just combined to build the very authentic layer 5 pyramidal cell. And then we add passivity, conductance, and dendritic capacity. They are pretty much general value of the model cells. And then we use the resistance 100 omega ohm centimeter with the base electrical meter. This is also pretty much general one. But for spine head and spine neck, we use Jim McGee's Nature papers value 385 ohm centimeters. And then this is we inject the IPSC current, and then using our trace itself. And then estimated duration and rise time and decay time constant, they are obtained from this, our practical real value. And then we did inject the septic current onto the SP1 spine head located here. This is like 34 micromillimeter from zone. And then this is the result. This is actually called the P. So this is current, but this is voltage. We inject this current in the spine head, and the spine head respond like this. So only spine head will respond very nicely, but this goes to the spine neck and also base dendrite. It's really reduced the size. And then soma, it goes to like really noisy level. So it actually doesn't really bother the soma at all, but only spine head is affected. The inhibition is really affected in spine head. Like this. And then I told you like whichever spine head is found to be innervated by inhibitory straps, they have the excitatory straps in the other side. So I just combine excitatory input together with inhibitory input. And the excitatory input is, I also use a general value, 0.2 nanoseconds. And then inhibitory static input with 0.1 nanosecond, which we obtain from our experiment. And then we just make a simultaneous injection of the excitatory and inhibitory, and it is really reduced. It's shown the green trace, it's really reduced like this. So it's at least this probably reduced the NMDA current, NMDA response. And then actually Higuri showed a very nice paper in the Science 2 years ago using the double uncaging method in the glue, uncaged glue and Ruby GABA, which is also uncaging GABA. And then he uncaged at the same time at the time, and then he shows the only glutamate uncaging of spine head 1, and he gets this calcium response. But combined with the Ruby GABA, it's really reduced into the half. It's quite similar trace like this. But the other one, I presume they don't have inhibitory static input onto this spine. Because my 2007 Journal of Neuroscience paper tells that about 2 to 3% of the spine, total spine population in the cortex, they receive inhibitory as well as excitatory, but the rest of them all, like 97% of the spine head, they only receive excitatory. And then we did also like a dendritic one, and we found quite similar effect. But interestingly, this dendritic inhibition also hyperpolarized the spine, but it's exactly same value. And in comparison to the spine head inhibition, it's really small value. So even though the base of the dendrite, it doesn't really order the spine head. So spine head is only hyperpolarized, inhibited by spine inhibitory static contact. And also we did somatic steps analysis simulation. And this is only one case, S1 is activated like this, and then we get this response of soma, which is 0.4 millivolts, which is not so big. Maybe it is not sufficient to block the spike initiation. But because of these guys large, and then they probably had a large probability, big probability of the release. And then I assume they are probably simultaneously activated. And so we just combine all of those steps activated together. And then we found that is like 1.4 millivolt that inhibited the soma. This really high inhibitions, and then probably block the spike activity of the pyramidal cell. And then this value is quite equivalent to shown by Professor Kudo and Shomoji, and they show that hippocampal basket cell inhibits pyramidal cell using the slice preparation and Purpose Nature paper. This is quite same value. So our simulation is quite authentic, I believe. And then this is just a plotting the IPSP peak of soma, and the somatic peak, somatic receptor inhibition peak is larger, but this part, dendritic and spine peak is really small after the conduction to the soma. So the only somatic inhibition really affects the soma activity itself, but dendritic and spine inhibition doesn't affect the somatic activity, but they only inhibit the local areas. So this is my a sort of summary. Somatic synapse is large conductance, and the EPSP is effectively blocked. This control neural output. And the dendritic synapse is small conductance, and the EPSP nearby and conducted EPSP from dendritic distal end is effectively blocked, but it does not hyperpolarize somatic membrane potential effectively. And spine synapse is very small conductance, and then it only hyperpolarizes spine head membrane potential. This has a strictly local betal. Now, maybe you may realize that then only somatic inhibition is necessary. And then maybe dendritic spine inhibition is they are not really big issue for the pyramidal cell. But that is not true because this distribution of the contact site tells you the answer. So these are the dendrogram, and then bar showing the contact site, and then this red is showing the real septic junction I confirmed in the EM study. But blue one, I haven't done the EM study, but at least light microscopic level, I confirmed their contact site. And then we know the three pairs showing the somatic inhibitions. And then whichever we found, the somatic inhibition is really clustered. In this case, it's 13 somatic steps only by like in baskets. And the other one, in this case, CS56 4, and then CS23 is I think anyway, other case, CS23 is like it's happened 4 or 5, but we haven't done the EM investigation. It could be much larger. But the other pairs, which only show the dendritic synapse, that is like this. Always, they are not really clustered. They are really scatteringly distributed. This means at least this branch is inhibited by the inhibition of this preservative basket cell. But also this one, this dendritic branch, this dendritic branch, this one, they are really inhibited. And then the excitatory input, maybe they just excite the post-synaptic pyramidal cell at the same time in a different part of the branch. And then that's the excitatory signal actually integrates through the dendritic branch, and then it becomes really big and strong. But all of those different branches really reduce the excitatory potential, and then it turns out the summation, the integrated EPSP should become much smaller. Then the dendritic inhibition also have a really big effect. This is also proved by the French women. Again, I forget the name of her, but she published in the neuron papers, and she just inhibit the activity of the OLMN cell, which correspond with the dendritic targeting cell in the hippocampus, and then that slice really get epilepsy conditions. So the distal dendritic inhibition is also very powerful inhibition. So this is my conclusion. Somatic synapse is large conductance and clusters, and then integrated EPSP is effectively blocked. This control neural output. And dendritic and spine synapse is small conductance, and the not cluster dispersed distribution and the EBSP on the dendritic branch must be weakened, and consequently, the integrated EBSP should be weakened. And then, now another question comes out. This is just a diagram showing our present experiment. So the different pairs are showing the different innervation pattern, like highly innervated soma or sparsely innervated soma, only dendritic innervation. But I wonder, maybe if this first spike basket so innervate, the other one is the same maneuver for them. And then I actually tried to count the percentage of the basket terminals, and I found all the 9 pairs. They also show the high percentage of the basket terminals. So that tells you this fast spike basket cell depended on the target terminal cell. It may innervate highly innervation soma or sparse innervation soma, or maybe only dendritic innervations. So it probably depended on the network. Maybe this has some network, and this has another belongs to another network, and this may have another network. That's kind of the idea, IMBB. So the same fast spiking basket cell innervates different pyramidal cells in the distinct style in terms of the number and site of static contact. Depending on the functional pyramidal cell subnetwork and distinct patterns of activity during different states. This is just my discussion in the summary. Thank you very much. These are the contributors. Thank you very much.
So usually you have another paper you early in the DB in 2012 where you report clustering of inhibitory synapse cells on the dendrite of pyramidal neurons. This is the opposite to what you presented, where you said that inhibitory contacts are not clustered. So how do you explain the differences?
Sorry, the beginning part I couldn't hear, so could you repeat?
So there is a paper with you, Chen et al. In Neuron in 2012, reporting clustering of inhibitory synapses on the dendrite of pyramidal neurons. So you said here that there is no clustering of inhibitory synapses on the dendrites, whereas in that study you saw clustering of inhibitory synapses. So what's the difference?
I don't quite understand, but I guess this is like one-to-one. If you think about one-to-one connections, and then they are not really clustered, and dendritic innervation that's clusters, but they have many different pairs, and then that's your question?
No, so the data you reported today are only from pair to pairs.
Yes.
So from the same neuron, essentially, you don't see multiple synapses onto the same dendrite.
Right.
But there is clustering of inhibition. It's just not coming from the same presynaptic neuron.
Different probably.
Okay. But in that sense, if you do have this kind of clustering, how would that affect your conclusion? Because you don't have input only from a single inhibitory cell at a given time, right? You have multiple inputs. So maybe this local control is not local anymore if many neurons contribute.
I think, yeah, I believe so. So anyway, like pyramidal cell innervated by a whole different type of interneurons. Our latest concept is like maybe pyramidal cell, fast spiking basket cell innervate soma area, but if they do, they innervate basal dendrite, but up to maybe 200,000 or maybe 100,000. That's their main target. But dendrite is the main branch of cell is mainly targeted to dendrite, and also neurogliaform cell. They call layer 1 extended neurogliaform cell and then we really don't know how they functionally innervate it.
So, thank you very much for your
Thank you very much.